speaker today, Dr. Christoph Thaiss. So Dr. Thaiss performed his undergraduate studies in molecular biomedicine at the University of Bonn, Germany, and his master's in microbiology and immunology at Yale University and ETH Zurich in Switzerland. After a visiting scholarship at the Broad Institute of MIT and Harvard, he performed his graduate studies at the Weizmann Institute of Science in Israel. He then established his own research group at the University of Pennsylvania in the microbiology department at the Perelman School of Medicine. His lab studies the role of host-environment interactions in metabolic, inflammatory, and neurological diseases, with a particular focus on the role of the intestinal microbiota in the regulation of host physiology. So Christoph, thanks for joining us today. We're really excited to hear about your truly interesting work. And with that, I will turn it over to you.
Thanks for the kind invitation. It's really, really great for me to be here today. This is actually my very first contact with the Monell Center ever since I came to Philly a couple of years ago. So even though today I'm visiting from my laptop, it's great to have you guys just a couple of blocks away, and I'm looking forward to hopefully many years of interactions and collaborations. So let me bring up my screen here. And please let me know in case it doesn't come up properly.
Looks good.
All right, great. So another reason why I'm really excited about being able to talk to you today is because there's probably no better place to discuss exteroception. And what we see as in my lab as our mission is to decipher how all these environmental factors, including the ones listed here, such as circadian rhythms, nutrition, psychological stress, environmental xenobiotics, temperature, physical activity, and many others, how all these environmental influences shape human physiology. And the reason why we're so interested in this topic is because it's now that we're almost 20 years after the Human Genome Project was completed. And the Human Genome Project, without any doubt, has been transformative for science and medicine. But at the same time, we haven't been able to stop this very rapid rise in many of the major human diseases across the population, including neurodegeneration, inflammatory diseases, and metabolic diseases. And the reason, of course, is because even though many of these diseases have a genetic component to them, the large majority of decisions whether an individual will get a specific disease are probably made by their lifestyle and by environmental factors. And that's why we think there is such a critical role in the next 20 years to be learned in understanding how these environmental factors shape human physiology, and by shaping human physiology, change our individual susceptibility to disease, and what the specific mechanisms are by which these environmental factors shape physiological processes. And typically when we think about exteroception or the impact of these environmental factors on the human organism, we think about the nervous system and the immune system. And thanks to many decades of mechanistic research, including a lot coming from your institution, we have a pretty good understanding by now what the sensors are, what the molecules are, what the pathways are by which the nervous system and the immune system perceive the environment and relay some of this environmental information to the host organism. But we think in my group that there is a third major sensing system of the human body, which is the community of all commensal microorganisms that colonize the human body. And in contrast to the nervous system and the immune system, we know almost nothing about how the microbiome perceives the environment, what the sensors are, what the regulatory mechanisms are, and especially what the pathways are by which the microbiome relays this information to the host and contributes to sensation of the environment and basically the body's reaction to the environment. And this is exactly what we're most interested in my group. So for the purpose of today, I'll focus on one specific environmental factor, which is this one here, because I'm sure all of you will agree with me that nutrition has played a major role, and maybe the major role over the last few decades, not only in changing human lifestyle, but in influencing our susceptibility to the various diseases that we discussed in the beginning, including metabolic diseases, neurodegenerative diseases, and many inflammatory diseases. And when we talk about nutrition, probably 99% of the field is busy trying to understand what we should eat and how the type of food is influencing almost any aspect of host physiology and susceptibility to disease. Now, I would argue that based on knowledge that was accumulated in the field for the last 10 years, it's at least as important to understand when we should eat, in addition to what we should eat. And this goes back to work that was conducted around 10 years ago by a number of labs, including the lab of Sachin Panda, who performed an experiment, which is shown here on the left side, where basically mice were fed a normal diet or a high-fat diet, a very standard obesity-inducing protocol in mice. But in addition to the typical ad libitum protocol, what they did was to limit the time of the day in which these mice could eat the high-fat diet. So they basically had a short window of a few hours during the dark phase in which the mice were allowed to eat the high-fat diet. And the remarkable observation that was reported in this work is that this completely prevents the susceptibility of these mice to obesity and metabolic disease. Because if you look at this dark red group here, even though these mice are on high-fat diet, just because they have limited hours of the day during which they can consume the diet, they don't gain weight as the control does. And this is not because they eat fewer calories, because they actually eat the same amount of food as the control group. So even though caloric intake is exactly the same, there are no detrimental metabolic consequences because of this time-restricted feeding protocol. And then the group went on to try this in many different paradigms, different hours of the day, different days of the week, different dietary formulations. And basically what they found is that this applies across the board. So whichever dietary protocol you choose, whichever timing protocol you choose, the effect is almost exactly the same, which shows that there is a pretty strong and pervasive role of this time-restricted feeding in preventing metabolic derangements. I'm showing here weight gain over time, but they looked at many other metabolic variables. And most recently, this also seems to work in humans, because when you look at when humans consume their calories over the day, first of all, you can see that it's pretty chaotic. If you look at the orange dots, we don't really have a clear 3-meal structure in most cases, because basically it seems that these participants eat at random times during the day. And when this is squeezed into a more narrow range, which you can see here in the blue dots, which is after the intervention started, then we get, just like in the animal system, we get a number of metabolic benefits, looking at various parameters of obesity and metabolism, all of which seem to benefit from this time-restricted eating protocol here. And I think this is most impressive when we look at continuous glucose monitoring, which in this individual here really brings it down to normal glycemic levels, while before the time-restricted eating protocol, this individual had spikes into very high glucose levels over the course of the day. So this is pretty impressive. And of course, as you know, this has been adopted widely in the general population. There's almost an unlimited amount of recommendations what you can do to improve your metabolic system. These are just some of them. There is now probably the most popular one is the 16-8 method in terms of how many hours you can eat and fast. But there are, just like in mice, there are now in humans, there are various adaptations of this theme. Some people prefer the 5-2 diet, which is basically 2 days of the week caloric restriction, the rest of the week normal eating. And as you can see here, there are many other protocols that you can follow, including the warrior diet and the spontaneous meal skipping diet. So basically all of them have the same goal of implementing some version of this time-restricted feeding approach. And of course, it's easier for some people to adhere to one of those than others, but overall they all seem to have the same metabolic benefits. And if this is so powerful, then of course the main question that we're interested in is what is the mechanism behind these beneficial effects of time-restricted eating? And the first one that immediately jumps to mind and was pursued by the community first is that this is most likely due to a system which is called the circadian clock. And I'm sure many of you are familiar with this molecular clock system, which is basically composed of various different interlocking feedback loops of transcription factors, which are expressed in almost every cell of the body and not only regulate each other in a 24-hour cycle, but also regulate downstream transcription of many thousands of genes which we call the clock-regulated genes that are expressed in a rhythmic fashion. And what's interesting about this circadian clock system here is that not only has it an intrinsically continuing rhythm when taken into isolation, but it's also highly adaptable and entrainable to the environment, including the light-dark cycle as well as times of food intake. So based on this, the molecular clock was an immediate, very attractive candidate for mediating the beneficial effects of time-restricted eating on systemic metabolism. However, there was another very interesting experiment performed by the group of Sachin Panda, which is shown here, where they basically asked exactly the same question of what is the role of the circadian clock in the metabolic benefits of time-restricted feeding. And they used various knockout mice that are deficient in some of these factors that you can see here. They use different knockouts for a clock, for BMAL1, for the cryptochromes, for period genes. They basically disrupted this clock cycle at various points of the feedback loop. And surprisingly, in all of these, the protocol still works. So if you perform these time-restricted feeding experiments on these clock-deficient mice, just like their wild-type counterparts, they have a strong metabolic benefit from having a restricted window of hours during which they consume the high-fat diet. So it seems like the protocol still works if there is no circadian clock. So what is the mechanism by which this time-restricted feeding works both in mice and in humans? So a number of years ago, we hypothesized that there might be a role for the gut microbiome in mediating the beneficial effects of rhythmic food intake. This goes back to when I was a PhD student in the lab of Eran Elinav, where we performed the first studies and is now followed through by a very talented scientist in my lab, Lenka Donalova. And basically the big hypothesis that is the basis of this large project is that we think that the microbiome in both its luminal communities as well as epithelial associated communities in the GI tract plays a role in mediating between the environment, which is inherently rhythmic because of the rotation of the Earth on its axis, and this intrinsic timekeeping system that I just introduced of the molecular clock, which is present in every cell. So the way we approached this first a number of years ago was by characterizing the microbiome by community analysis, but on a resolution which had never been done before, which is looking at every 6 hours over the course of 48 hours. And to our surprise at the time, what we found was that many taxonomic elements of the microbiome, including the ones shown here, actually show a very robust circadian oscillation across the course of a day. So you can see here that they don't all have the same peak and trough times, but they actually have unique oscillations that are not completely synchronized, but seem to go in waves during the community. And here you can see on the taxonomic level of the entire microbiome by 16S sequencing that we have these oscillatory waves going through the community over the course of 48 hours. Now, as I said, this was looking at the community by 16S sequencing, which we use to profile the taxonomy. But on the same DNA, we can perform whole genome sequencing to resolve the entire genome of the microbial species that we find. And when we do this, we can apply an algorithm which is called the Jtk Cycle Algorithm, which is typically used to score rhythmic elements in large-scale data sets. And then we can choose a Q-value cutoff, and we call everything below this cutoff, we call this a rhythmic element. And you can see here in this heat map that these are indeed highly rhythmic, basically by looking at this red-blue pattern across 48 hours, you can see that these are very robust circadian oscillations. And when we have a map like this, what we can do is we can go in and we can assign functions to these different genes that are rhythmic. And when we do this, we can see that, to our surprise, many of the metabolic functions performed by the microbiome don't seem to be stable across the course of a day, but are actually undergoing the same rhythmic pattern, which you can see here as this red pattern across 48 hours. And you can see here that this affects many of the really major metabolic functions of the microbiome, including nutrient biosynthesis, anabolic and catabolic reactions, as well as detoxification reactions. So from this, we learned that the microbial community is not unlike what we had assumed before, is not stable over the course of a day, but actually undergoes these hour scale fluctuations. And this is actually not only true in terms of the composition and function of the microbiome, but even the localization of the microbiome, which we first found by electron microscopy studies of the epithelial adherent communities in mice. So here we're looking by electron microscopy at the large intestine of a mouse at 6 p.m., which is when the mouse, basically when the lights go off in the animal facility and the mouse starts eating and or wakes up and becomes active. And you can see that the community of adherent bacteria is relatively sparse. But as we go into the night and the mouse becomes active, the communities become a lot denser and a lot more localized to the intestinal epithelial surface. And this basically continues until the early morning when the lights go on again and the mouse starts becoming less active and starts sleeping. And then during the light phase, we're back to a very sparse epithelial community. And we did this in many iterations and quantified the results. And you can see here that the number of bacteria per centimeter of tissue is actually undergoing an almost log-fold difference between the dark and the light phase in a very reproducible circadian pattern. And what we can also do is we can take these epithelial adherent communities and again, subject them to 16S taxonomic sequencing. And we see that not only the amount of bacteria that are in contact with the epithelial cells is highly oscillating, but also the taxonomic composition of which bacteria there are is not stable across the course of the day, but is undergoing these fluctuations. And you can see this here based on on beta diversity quantification. So now we know that the community composition, its function, as well as its localization in the GI tract seems to be undergoing these circadian oscillations. So now the next logical question is, what does this mean in terms of function for the host? And to get there, I first want to introduce yet another layer of circadian oscillations, which is looking at the functional output of the microbiome in terms of its metabolome. So here, what we did is we performed an unbiased metabolomics experiment of all the molecules we could quantify in the lumen of the GI tract across 48 hours. And again, you can see that many of the molecules that we find in the GI tract, both those produced by the bacteria, as well as those produced by the host, and many of those that are produced by both or coming directly red-blue pattern across the course of the day. So many of these molecules that we know are very important for host physiology, including serotonin and some of the other molecules that the microbiome is involved in producing, are not stable over the course of the day, but they undergo these fluctuations. And since we have a paired data set of the metagenome and this metabolome, we can go in and we can reconstruct entire pathways, such as the microbial conversion of ornithine to proline, as well as the microbial production of biotin. We can see that some of these pathways are oscillating at each step along the pathway, including both the abundance of the biosynthetic enzymes, as well as the abundance of the resulting molecules themselves. So based on all this, we can conclude that the entire ecosystem in the gut is very rhythmic, and we can reconstruct what we call a day in the life of the microbiome, which is basically the bacterial counterpart to the host circadian clock, where we can assign specific hallmark species and specific hallmark metabolic functions or metagenomic functions, and even specific hallmark metabolites to distinct times of the day. And in all the experiments we've performed so far, we see this we see that this is a very stereotypic pattern that happens across different animal facilities, across different strains of mice, across different microbial community composition. So it seems to be a very conserved system that seems to be very robust and always affect the same type of species and their metabolites. Now, one question we had was, do we see any phenomenon like this in humans? Because in mice, of course, we have a very strict activity cycle. The lights always go off at the same time. The mice always eat roughly at the same time. So can we see any microbiome fluctuations of the same sort in humans? And luckily, we were able to obtain microbiome samples across multiple days from a number of individuals who donated stool samples for analysis. And we can see that in these individuals, we see the same robust fluctuations, in this case, even across 5 days, in terms of microbial species oscillations. And we can see the same phenomenon on the level of metagenomic function if we do shotgun metagenomic sequencing, where again we have some of the same metabolic pathways of the microbiome not being stable across the course of a day, but undergoing these time of day dependent fluctuations. And very recently, in a very nice study coming from the lab of Dirk Haller, they expanded this concept to a large number of humans in what they call the CORA cohort, which is a cohort of several hundreds to thousands individuals, in which they basically collected the microbiome and they noted the time of day in which the microbiome was collected. So they were able to reconstruct almost a full 24-hour cycle based on when the microbiome sample was obtained. And you can see here that they basically had a pretty good coverage of almost all hours of the day in terms of when the samples were obtained. And then interestingly, when they reconstructed the time of day in terms of when they detected different features of the microbiome, either alpha diversity or different species abundances, they were able to see in many operational taxonomic units, which is a proxy for species, that these were undergoing these circadian rhythms across the day. So this is something that is not only happening in mice, but it seems to be happening in humans and can be seen even at a very large scale of hundreds and thousands of individuals in a cohort study. So now that we know all of this, we can come back to our original questions and ask what is the function of these microbiome oscillations for host physiology, or are these microbiome oscillations functionally meaningful? And here I want to take a small tangent and introduce you to some of the major known function of the microbiome, as has been studied for the last 2 decades or so. And among the many functions of the microbiome, I want to highlight 3 especially, which I think are the ones that will be most affected by the fact that the microbiome is a rhythmic community. Of course, I'll talk mostly about nutrition because this is why we started the project. But just as a small tangent, I want to mention that there is probably a very strong effect of the microbiome being rhythmic on its ability to deal with pathogenic invasion and environmental xenobiotics. And the reason why I say this is because we know that some of the primordial functions of the microbiome are related to providing colonization resistance against pathogenic invasion. So the microbiome is extremely good at excluding foreign members of the microbial community, which actually helps us a lot fight against enteric infections like Salmonella, because the community is basically occupying metabolic and spatial niches that make it very difficult for invading pathogens to establish themselves in the GI tract. And now that we know that the microbial community is actually rhythmic, this means that probably this colonization resistance is not a stable function across the time of the day, across the 24 hours of the day, but is actually undergoing rhythmicity as well. And this is something we're currently looking at into more detail. The same is true for xenobiotics. We know that the microbiome is a major contributor to drug metabolism in the GI tract, and actually has a strong influence. It's now known for many different examples of drugs that the microbiome has a very strong influence in determining the efficiency and toxic side effects of various drugs. So again, knowing that the microbiome is not a stable community, but a rhythmic community, this would also mean that probably this xenobiotic degradation or drug metabolism function is also rhythmic across the course of the day. And this brings us into a very interesting recent field, which is called chronopharmacology, which is basically thinking about the fact that in the large majority of cases, when we're prescribed a drug from a physician, it's only in the small minority of cases does this come with a recommendation as to the time of day when to take the drug. In the large majority of cases, it's almost up to the patient to decide when to take the drug. And we know from many detailed studies that the time of day is actually a very strong influencer of drug metabolism and drug efficiency as well as toxic side effects. So we thought, can we look at the possible role for the microbiome in chronopharmacology? And I'll just show you one piece of evidence before I go back to nutrition, but I want to highlight the point that I think there does seem to be a very strong effect here. And we applied this in the case of a mouse model of paracetamol overdose, which, as you know, is one of the major causes for acute liver failure. And the same happens in a mouse. When you overdose a mouse with acetaminophen, you get very strong liver damage, and this was known from the literature to be very circadian. So it seems there is a very strong effect of the time of day in terms of the amount of damage that acetaminophen overdose does to the liver. And we recapitulated exactly the same. If you just focus on the blue graphs here, ZT0 is sort of the circadian jargon for the beginning of the light cycle. So this is when the lights go on, and ZT12 is exactly 12 hours later. So this is when the light goes off. And you can see that depending on the time of day in which we overdose a mouse with paracetamol, liver damage in terms of ALT and AST release, as well as histology score, is much greater when we come at the end of the light phase. Now, what's interesting is that when we do the same experiment without the microbiome, either by depleting the microbiome using broad-spectrum antibiotics or performing it in a germ-free facility, this time of day dependency is no longer there. So it seems like the microbiome is the contributor to this time of day dependent liver damage caused by acetaminophen overdose. Now, what's really interesting to explore is whether this is because of the microbiome's effect on the liver or because of the microbiome's effect on the drug. And we're exploring this now, not only in the case of paracetamol, but many other examples of drugs that are probably following the same principle. But now, with this small aside here, let me go back to nutrition and how the whole thing may relate back to time-restricted feeding and its metabolic benefits. So we started looking at this in the context of a jet lag model, which is a very common way of disrupting the circadian clock of mice. In our case, we're basically exposing them to an 8-hour time shift every few days, which is the mouse model for flying between Philadelphia and Dubai every couple of days, because this is exactly an 8-hour time shift here. And when we do this, we basically recapitulate what's known from the human epidemiological literature, where we know that people with a disrupted circadian clock, like shift workers and others that are frequently disrupting their own circadian clock, we know that this induces metabolic derangements in humans, including higher susceptibility to obesity and diabetes. And it seems like we're seeing the same in mice, because if you just focus on the black and the red group here, which is the control group and the quote unquote jet lagged mouse, you can see that the jet lagged mouse will gain weight over time when kept on a high fat diet, will become very glucose intolerant, and will accumulate body fat content. And at least in this system, again, this seems to be completely dependent on the presence of the microbiome, because if we come again with this broad spectrum antibiotics protocol, and we treat both control and jet lag groups, they are no longer susceptible to weight gain, and they are no longer susceptible to hyperglycemia, and they don't accumulate body fat content. So it seems like this the detrimental effect of this clock disruption in the jet lag system is dependent on the microbiome. And we can even do the reverse experiment, where we take the microbial communities from control and clock disrupted mice, transplant them into germ-free mice, and look at the metabolic consequence for the recipient. We can see that the recipients basically mirror the donors in terms of their susceptibility to weight gain, glucose intolerance, and body fat accumulation over time. And again, going back to this very nice study from Dirk Haller's group, they expanded this concept to a large-scale human cohort, because in their cohort, they not only looked at healthy individuals, but also at pre-diabetics and type 2 diabetics. And they saw that these microbial species oscillations in the GI tract are largely gone in the pre-diabetics and the type 2 diabetics. So this community rhythmicity is a feature of health, actually, and it's lost in metabolic disease, just like we saw in the mice. And what's even more important, I think, is that this loss of species oscillation seems to be an independent risk factor for developing diabetes, because since they covered quite a few years in their study from 2013 to 2018, they were able to follow a couple of individuals that went from being non-type 2 diabetic to pre-diabetic and then type 2 diabetic. So in those individuals, what they were able to do is to see whether the loss of species oscillations in the gut would be an independent predictor of their susceptibility to develop type 2 diabetes. And this is what you can see here on the right, that just by looking at species oscillations by themselves, you can predict an individual's susceptibility to develop diabetes over the course of these 5 years. And this is only marginally improved by adding BMI, which shows that the species oscillations are largely independent. All right, so now we have all the ingredients we need to go back to the major question, which is, what is the connection of microbiome oscillations and feeding times? Because this is basically where we had started the project, and which is most interesting in terms of understanding the metabolic benefits of time-restricted eating. So to approach this question in the first place, we did an experiment where we limited the amount of not the amount, but the time at which the mice can eat, very similar to this original experimental paradigm that provides all these metabolic benefits. But in this case, we're basically alternating, not in the same group, but between two groups, we're alternating the time of day in which the mice can eat. You can see here that we can do this, of course, very efficiently in metabolic cages by restricting food access to 12 hours, either during the dark phase or during the light phase. And then we came back with our microbiome profiling approach, and we characterized the microbiome over the course of 48 hours to see what happens to community oscillations when food intake is limited to these different times of day. And to our big surprise, we keep overall oscillations in this approach, but we actually completely shift microbiome oscillations by 12 hours. So here we're looking at one very prominent species in the microbiome, but the same is true for any other species we looked at, which is that the oscillatory profile is preserved, but is now shifted by 12 hours depending on whether the food comes during the dark phase or during the light phase, which means that we can completely control these oscillations by controlling the time of food intake. And then reversely, if we can control the time, of food intake and thereby control microbiome oscillations, what happens in an animal that doesn't feed rhythmically? So we went back to this scheme of the circadian clock, and we used animals deficient in one of these different members, which we know makes the mouse basically confused in terms of the time of day. So they start eating in an arrhythmic fashion. And we're doing this here, in this case, with period 1-2 double knockout mice. So these are arrhythmic mice, clock deficient mice. And now we come back and we profile the microbiome over the course of 48 hours. And now you can see, now we lose species oscillations. Again, as you remember from this Jtk cycle analysis, everything below this dotted line would be considered a rhythmic species. And in the wild type, which is the blue one, we have plenty of rhythmic species down here. We basically lose almost all of them. And here is one example of them in the knockout. So basically having a non-rhythmic host also renders the intestinal microbial community non-rhythmic. And now the question is, if we restore rhythmic feeding, can we bring back microbiome oscillations in this mouse? First of all, I want to show you that not only are the species oscillations affected, but also the metabolic oscillations, because if we do shotgun metagenomic sequencing on the period 1-2 double knockouts, now you can see that all these metabolic functions and KEGG pathways of the microbiome that used to be rhythmic in a wild type mouse are converted into a random non-rhythmic pattern in the knockout. So their entire community is truly arrhythmic in terms of all the functions that the microbiome performs. And now we do the experiment that I just told you about, which is to bring back rhythmicity in the microbiome. So here we're just first, we quantify at baseline in ad libitum fed mice that species oscillations are really reduced in the period 1-2 double knockouts, which you can see again here. But when we restore rhythmic feeding by either feeding them exclusively during the dark phase or exclusively during the light phase, now we can bring back the amount of oscillating species in the knockout almost to the level of the wild type. If we look at their oscillating profile, we can see that, again, depending on whether the food comes during the dark phase or the light phase, the microbiome oscillations in these major members of the microbiome will be shifted by 12 hours. So not only can we restore the oscillatory properties themselves, but we can again direct their peak and trough times to specific times of the day, depending on when we feed the mice. So now the question is, how does this relate to the metabolic benefits of time-restricted feeding? So to answer this question, we set out to do a very large-scale study in which we took different groups of mice, controls that are fed completely ad libitum, and then an intervention group in which we first have ad libitum feeding at baseline, but then we perform again light phase restricted feeding. So we basically switch the mice from having a feeding pattern where they voluntarily eat the majority of their food during the dark phase to a 12-hour inverted feeding pattern, which we then maintain for about 10 days before we switch them back. And during this time, we not only control their food intake, but we monitor their activity, their energy expenditure, many different metabolic parameters. And of course, we look at the fecal microbiome to understand how this very precise feeding intervention would change the microbiome and influence its metabolic consequences. So first, what I want to show you is what this looks like in terms of the microbiome oscillations as we go into this shift in feeding patterns. So I showed you already that when we wait a number of days and we profile them back here, the microbiome community is shifted by 12 hours in terms of their oscillations. But now the interesting question is, how do we get there? What happens as we shift their feeding times and as we wait for this inverted pattern to be established? And this is actually where it got really interesting, because if you look here by different parameters of alpha and beta diversity, so this is basically the an index for community richness, and this is a component of the principal coordinate analysis that we perform to profile beta diversity. You can see that initially, as we disrupt the feeding pattern, the oscillations are completely lost, and it takes about 5 days for them to be reestablished. So when we acutely disrupt the feeding patterns, microbiome oscillations are lost. The microbiome is basically confused as to this oscillatory pattern that is established by rhythmic food intake. And then after 5 days, we regain a very robust oscillatory pattern. And you can see that the same is true here as well on the level of beta diversity. So basically by shifting the time of food intake, we establish this 4 to 5 day period of loss of oscillations. And you can see that this applies throughout the entire community. Here we're looking at a specific taxonomic element, but we can quantify this for all the different oscillating OTUs in this case, where you can see that we basically the number of oscillating OTUs initially plummets when we do this feeding intervention. Then it takes them a couple of days to come back to their original level, and then it stays stable, because as you remember from the previous experiment, it doesn't matter if the food comes during the day or during the night. The amount of oscillating species will be roughly the same, just the phase of the oscillation will be different. And here you can see what happens in the transitionary phase, which is that there is an acute loss of oscillations and then a regain of oscillations. And if you think about it, this is actually what likely happens in humans that are disrupting their own circadian clock, because they either perform shift work, or they stay up late at night, or they have irregular working times. Because basically and you remember from the plot that I showed you earlier from Sachin Panda's paper, that this is basically what humans do all the time. They don't follow a very strictly rhythmic meal pattern over the course of different days, but they actually eat relatively chaotic when it comes to the time of day. So we think that this probably, just like in mice, provokes this loss of rhythmicity in the intestinal microbial community. And now what I want to show you next is what are the metabolic consequences of this transient loss of species rhythmicity. And here we had first evidence by actually looking at various time points of this experiment in terms of metabolic consequences. And I'll just show you the data for the glucose tolerance test, but the same is true for many other metabolic parameters. So if we look during the disruption phase and we compare the control and the time feeding group here, you can see that the time feeding group becomes acutely glucose intolerant. So it seems like during this phase of microbiome confusion, if we may call it microbiome confusion in terms of the time of day, there is acute hyperglycemia in terms of when we come with an oral glucose bolus. And if we wait longer and we follow the mice further into the experiment, this completely goes away. So after a couple of days, there is no longer hyperglycemia. And in fact, you can see already a slight trend towards a lower glucose level here. And if we follow the mice for much longer and we keep them on this time-restricted feeding paradigm for much longer, then we can see what's known in the literature, which is that this is actually beneficial for glucose levels. But acutely, we have this, as we have this uncoupling of feeding times from microbiome rhythms, we have this strong susceptibility to hyperglycemia. So we thought, is this actually dependent on the microbiome, or is this because of this acute disruption of feeding times? So when we do exactly the same experiment, but again, we're depleting the microbiome using broad spectrum antibiotics, there is no longer susceptibility to hyperglycemia. So it seems like this initial hyperglycemic phase here is mediated by the microbiome. And if we now do an approach which is, again, probably more relevant to the human situation, where we keep the feeding disruption going, so instead of doing a simple, basically one time point switch to opposite feeding times, we keep switching the mice every 3 days, like a shift worker would do that changes shifts every 3 days. And we basically do daytime feeding for 3 days, nighttime feeding for 3 days, and then we switch to daytime feeding. And we keep this going, then we have chronic hyperglycemia, which never reverts back to normal. So if we perform these chronic changes in food intake, then we actually move the mice into probably a pre-diabetic state, which is very similar to what we see in shift workers and other individuals that experience chronic disruption of their circadian clock. So finally, what I want to share with you is how we can explore the mechanisms underlying this microbiome-mediated susceptibility to metabolic derangement in this system. So what we did to start exploring this is we performed unbiased RNA sequencing of the intestinal epithelium across 48 hours to score circadian expression, this time not of the microbiome component, but of the host component of the GI tract. So basically what we did here is we took intestinal epithelial cells every few hours for 48 hours, and we scored transcript abundance, again, using this Jtk Cycle Algorithm, and everything, this you know by now really well, everything below the dotted line is a rhythmically scoring element. And then at the same time, what we did is we performed the same experiment in microbiome depleted and control mice. So now we can compare this rhythmic oscillatory pattern, not only in the natural setting of microbial colonization, but also in the absence of the intestinal microbiome. So we can take these rhythmic elements here, and we can look at the genes that are influenced by the microbiome. So first of all, we'll find a few of the known candidates here, like the transcription factor RORC, which is known to be rhythmic and known to be influenced by the microbiome. So this was a good positive control. And at the same time, we find genes like GCG, which, as you know, encodes for the precursor of GLP-1. So we can basically see that this precursor of GLP-1 is not only highly rhythmic, but also strongly influenced by the microbiome. And since we know that GLP-1 is such a major driver of glucose tolerance and regulation of insulin release, now we hypothesize that perhaps a potential molecular explanation for this is microbiome-induced rhythmic expression of GLP-1, which is then adapting the glucoregulatory system of the host to the times of food intake. So basically, to cut to the bottom line here is we can use various pharmacological interventions. I'll just show you one for the sake of time with the GLP-1 signaling, in which we use liraglutide, in this case, a very strong and actually clinically relevant pharmacological agonist for GLP-1 signaling. You can see that in the vehicle control, again, we have this time feeding group and the control group, and we get this microbiome-dependent acute hyperglycemia a few days into the disruption of food intake, or time disruption of food intake. But when we do this in mice that are receiving liraglutide, just like the mice that are treated with antibiotics, they are no longer susceptible to this microbiome-mediated glucose intolerance. So we think that potentially one of the mechanisms by which the microbiome is mediating this adaptation to timed food intake is by activating GLP-1 signaling, and thereby preparing the host for glucose to come down the GI tract and basically metabolically prepare the system for food intake and for glucose tolerance. So I'll summarize before thanking everybody who was involved in this project. I'll summarize what I've shown you so far. So we think that regular feeding times are extremely important in establishing this microbial community rhythmicity. While when we have abrupt changes in feeding rhythms, which we can do experimentally in mice, but we as humans actually do voluntarily all the time, we have a negative consequence for this oscillatory pattern of the microbiome. And this is probably due to, even though we don't understand the exact mechanisms by which this works, this is probably due to rhythmic nutrient availability coming down the GI tract, and then basically being used in a cross-feeding network within the microbial community, where one species of the microbiome directly feeds off dietary nutrients, and other species feeding off nutrients released by those primary microorganisms that establish this rhythmicity in the GI community. Now, what's most important in terms of metabolic health of the host is that this seems to affect not only the GLP-1 system, but many other metabolic systems that are involved in glycemic control. And while we have these robust microbiome oscillations, we have proper glycemic control as long as the time of food intake is synchronized with microbial community oscillations. And once we have a mismatch of food intake times and loss of microbial oscillations, then we have impaired glycemic control, probably because of deranged GLP-1 signaling and potentially other pathways. So now the reason why this, I think, is important is because now we have a completely host circadian clock independent explanation of why time-restricted feeding would be beneficial. Because probably any of the protocols that we used in mice and we see used in humans in terms of limiting the amount of hours during which you consume food, they will probably have very beneficial effects for microbial community oscillations. Because anything that induces rhythmicity in food intake habits will induce rhythmicity in the microbiome. So whether you're following a 8 to 16 hour protocol, or an every other day fasting protocol, or a 12 hour, or you name it, like any combinations of hours that you can control your food intake during those hours, this will induce rhythmicity in the microbial community. And rhythmicity by itself is probably a very strong feature which helps maintain glycemic control through their impact on the intestinal epithelium. And I think that is exactly the reason why in this cohort study from Dear Colors group, they found microbial oscillations to be an independent risk factor for developing prediabetes and later type 2 diabetes. Because no matter what you eat, as long as it comes rhythmically, you will see microbial species oscillations, and these have metabolic benefits in terms of regulating GLP-1. So we can basically independently control not only what we eat, but when we eat. And I think both the nutritional composition as well as the timed intake of those nutrients are basically separate risk factors that we shouldn't mix up in terms of predicting individual risk for diabetes and prediabetes and potential therapeutic interventions. One last point I will make is that now that we understand this system so much better, we can probably also think about pharmacological interventions downstream, because as I mentioned, the GLP-1 system is pharmacologically extremely well studied. And we know that the microbiome affects even other components of this glycemic control system. So I think one potentially very attractive consequence of this is that we can not only restore rhythmic feeding by adhering to some of these time-restricted eating protocols and making our microbiome rhythmic again. But we can probably leverage the beneficial effects of these oscillations by optimizing chronopharmacology when we think about interfering with GLP-1 or some of these other signaling mechanisms. Okay, I think I'll stop here. So we have enough time for questions, and I want to thank many of the individuals that were involved in this work. As I told you, the whole project started when I was a PhD student with Aran Elinav, and we collaborated extensively with Aran Segal at the Weizmann Institute. And now this project is spearheaded by Lenka Donalova in my lab, who is a fantastic scientist, and she's helped by many other members of the group that I'm very fortunate to work with. And this work is supported by several funding agencies that I thank for making this possible. And thank you again for inviting me today, and I'm looking forward to the discussion.
Thank you so much for a really fascinating talk. You can unmute and jump in and ask questions. You can type them in the chat or direct message me and let me know you have a question. And I think I'll get the discussion started with what might be a big question. So I think you've shown your work, beautiful work from you and others in the field, a really strong relationship between the gut microbiome and metabolism. But as a neuroscientist, I think of the brain as the master regulator of metabolism. So my question is, what's the mechanism? So are these effects kind of peripherally mediated? Is there a signaling pathway to the brain? Both? None?
Yeah, no, this is extremely interesting. And to be honest, the microbiome field has undergone, or is still slowly undergoing, kind of a shift in how we think about potential mechanisms by which the microbiome regulates metabolism. So when the first studies were done more than 15 years ago by now, where Jeff Gordon's group at WashU and later many others realized that the microbiome has such an important role to play in metabolism, it was very focused on the endocrine activity of the microbiome, because we know that the microbiome is such an important source of metabolites that gain access to the bloodstream and then gain access to the liver, to adipose tissue, to muscle, and many other of the peripheral metabolic organs. And this is where basically almost all the attention was focused on, in trying to understand these metabolite circuits of how the microbiome contributes to peripheral metabolism. And only in the last few years have we started understanding that probably a lot of the metabolic secrets of the microbiome are to be localized in the brain. And there is now, as you're well aware, there is a very strong interest in understanding the signals, the microbiome control signals coming from the gut and influencing metabolic decision-making and metabolic behavior in the brain, including control of food intake. And I think in the case of our study, GLP-1 is actually a very good example of a very well-known mediator that influences the brain and thereby controls systemic metabolism, which is actually very strongly controlled by the microbiome. This was a big shock to the community initially when it was realized that GLP-1 is such a strongly microbially driven molecule. And actually, in the absence of the microbiome, GLP-1 levels are extremely high. So the microbiome is inhibiting the release of GLP-1 from enteroendocrine cells. So I agree with you that the field of metabolic host microbiome interactions should think a lot more about the brain, and it may turn out that many of the mechanisms that have been described early on in terms of the microbiome's ability to influence weight gain and obesity and diabetes are mediated by the brain, but many of the mechanisms are still completely in the dark.
Just a quick follow-up to that is, so I could see, the microbiome influencing these incretin hormones and then influencing the brain. Is there any knowledge of a more direct signaling pathway for the gut microbiome to the brain, or is it all thought to be mediated through hormonal mediators?
Yeah, this is all very recent stuff coming out of several labs across the world, where we're starting to understand how the microbiome is not only influencing the activity of enteroendocrine cells, but in some cases even directly influencing the activity of afferent sensory neurons. Cool. So there are both ways, both basically direct and indirect ways, using enteroendocrine cells and maybe other epithelial cells as a relay system by which the microbiome is is influencing the sensory nervous system component that innervates the GI tract. Of course, we know that the vagus nerve and also the spinal cord afferents, they have terminals in the GI tract. So the GI tract is along its entire length, as you well know, is very densely innervated. And in terms of the microbial influence on the nervous system, this was for a long time. This was exclusively focused on the enteric nervous system, on the resident neurons that have their somata in the myenteric plexus. But for the last few years, so many examples have unsurfaced by which we understand that the microbiome directly influences the activity of these afferent sensory neurons. So I think we can envision that not only nutrients coming down the GI tract, but also molecules directly produced by the bacteria can influence activities in the brain within the range of seconds, and don't need the circulation to be used for messaging to the brain.
Awesome, thank you. Other questions?
I have a question. Does the microbiome in the nasal passage, does that have a circadian rhythm? Because there they have direct access to neurons, at least viruses do, but I would assume bacteria do as well.
Yes, this is a very interesting question. There are actually two really interesting components to it. One is, what about the microbiome outside of the GI tract, right? Either in the nasal cavity or in the oral cavity or on the skin or in the lung or many other places where we know that commensal colonization exists. So what about the microbiome there? And the second aspect to your question is, what about, I've shown you only the bacterial community of the microbiome, but we know that there is a very strong non-bacterial community, including a commensal virome, eukaryotic communities, fungal communities, and so on. So for the first question, there have been a few studies looking outside of the GI tract, including human studies looking at the oral community in terms of are the species rhythmic, what controls microbial community oscillations there. We know from those studies, even though everything is pretty preliminary compared to the GI tract, but we know that rhythmicity exists because species oscillations have been found. We don't know at all what controls them. Is it food intake? Are there any other circadian environmental factors that would play a role? And it's even more unclear, which is what you're actually asking, what is the impact of on host physiology, either sensory neurons in those places or other activities in the different organ systems. So this is very early days, but something that is very interesting. The second component is also completely in the dark still. We only know that these rhythmicity features are affecting the bacterial component of the microbiome. We don't know anything about non-bacterial microbiomes and whether they're rhythmic, but this is something we're actively investigating.
I have a question.
Go ahead, Mark, and then Paul.
Yeah, I have actually two questions. One is a short one. The longer one is thinking of Peter Tarnbow's publication recently showing that a ketogenic diet affects the populations in the gut, the bacterial populations in the gut, apparently due to the ketone bodies in the gut. Have you looked at any other differences in diet composition, effects of diet composition?
Yeah, that's an interesting point. We have only done comparisons of normal chow diet and high-fat diet, and several other groups have as well. And the unanimous conclusion is that high-fat diet dampens or decreases the amount of species that are oscillating. So diet and dietary composition definitely has a very strong role to play. We haven't looked at ketogenic diet in terms of microbial oscillations yet, and I'm not aware of anybody else has. But if I had to make a guess, I would put my money on a very strong effect in terms of microbial community oscillations, because the nutrient composition of a ketogenic diet is so extreme compared to most other diets.
Yeah, it's certainly a high-fat diet, so you might answer the question whether it's the fat or something else about those high-fat diets that's having the effect. My short question is, do you see the same effects on glycemic control if you use an intravenous glucose tolerance test?
Yeah, that's a very, very interesting question. We actually have done this experiment and we don't see an effect there. So it seems like this is exclusively affecting glucose that comes through oral nutrition.
Right. Fascinating.
Which would explain why which makes sense in terms of the effect being mediated by the microbiome and intestinal GLP-1.
Yeah. Thanks. Lovely talk. Thank you.
Thanks.
Paul?
Yeah, I second what Mark said. That was a really, really lovely talk.
Thanks.
Thoroughly enjoyable. So I too have two questions, or sort of a comment and a question. My first is about the translational implications of this. So if the control is really about rhythmicity, it's not necessary to follow a Ramadan-esque pattern of just restricting your feeding to a brief period of time each day. You could eat 3 square meals a day, but as long as they were at the exact same periods of time every day, you would establish that rhythmicity. And if that's true, then really the problem comes in sort of arrhythmic snacking in between, like in that graph that you showed where people kind of just feed all day long. That's really where the problem comes in. Is that correct?
Yeah, that's a very interesting thought. We actually don't have an answer to this exact question. We thought about doing an experiment where we, as opposed to having a 12-hour feeding and a 12-hour fasting window in mice or in humans, where we have very strict rhythmicity, but food every 6 hours, just to see if rhythmicity of any shape or form would cause these oscillations in the microbial community and the metabolic benefits. We don't know if that's going to be the case. I would assume that a certain window of fasting is necessary for robust oscillations to be established, most likely because there is this, we hypothesize that there is this wave of cross-feeding events going through the microbial community. And I think if nutrients or food is introduced too frequently over the course of a day, then we might disrupt this cross-feeding mechanism from happening in the community. But it's something we cannot rule out at this stage. So I think I agree it would be a very, very interesting experiment to do.
And my second question is really one of the hormonal controls of weight gain and weight loss, irrespective of microbes. So I think you said both through antibiotics and through xenobiotic mice that the time-restricted feeding resistance to weight gain doesn't work in those animals. Is that what you said?
So we haven't done this very experiment. We haven't basically performed this time-restricted feeding experiment in germ-free mice, for example, and looked at the metabolic consequence of high-fat diet. I would agree that this would be a good next experiment to do. The problem is that with this experiment, we know that germ-free mice generally are less susceptible to weight gain on high-fat diet. So we would hypothesize that just by the fact that they don't have a microbiome, they already don't gain as much weight as the colonized counterpart. We've basically done the experiment that you're mentioning when looking at glycemic control, not high-fat diet-mediated weight gain in the long run, but glycemic control after a glucose tolerance test. And there we see exactly what you say, which is that the microbiome is the mediator of these beneficial effects.
Well, I was actually thinking it would go the other way, which is that there should be certain requirements of insulin and its role in putting weight on or allowing lipids to come out of adipocytes in the absence of insulin during feeding that should have nothing to do with microbes. That should be superimposed on top of everything else, and whether or not the microbes are there should make no difference there.
Right. I'm
Either
not
that
saying
or
that
we don't understand what insulin's doing.
Right. So even though what I should say is that even though we know that there is a very strong impact of the microbiome on regulating insulin release and glucose tolerance, from the animal experiments, we know that this is not the only component. Of course, there is a direct influence of dietary glucose, which directly reaches the circulation and regulates insulin. So there is certainly also a non-microbial component to it.
All right, thank you. Your talk was really lovely.
Thanks.
All right, well, we're about 5 minutes over, but I'm just going to ask one more really light question. And that is that you can buy a lot of these gut bacteria like on Amazon, or even, probiotics at the grocery store or CVS. So What do you think about that? Is this going to be something that people are going to want to use to regulate their metabolism, or is it going to just be more nuanced than that?
Well, to say the truth, the probiotics you can buy on Amazon or in CVS, they of course don't undergo any regulatory approval. So none of them are clinically proven to have any metabolic benefits or other benefits. So we're just lacking the studies to know whether these are beneficial on the large scale or not. My recommendation would be that if you take these probiotics, then at least take them rhythmically. So you introduce some rhythmicity by adding this external component, but the truth is that in all likelihood you will just find them in the toilet later on, and they don't establish themselves in the GI community.
All right. Well, thank you so much again for a really brilliant talk. We appreciate you taking the time. I will give you a little bit of break before your next meetings.
Okay, sounds great.